Hello dear students, welcome in class. I am Bhupinder Verma, your science teacher. So dear students, today we are going to start chapter 5 of class 9 science, that is cell structure and function, and say that cell is the fundamental unit of life. This is the first part of chapter, and in this section we discuss about cell and its related discoveries and its components. So first is, we all know that all living organisms in this universe are made up of cell. We know that the basic unit of every living being is cells. That's why we say that the every living being is made up of cells. That means like a wall is made up of bricks, in same sense a human or animal is made up of cells. So that's why cell is a basic and fundamental unit of living beings. Like atoms are the basic fundamental unit of matter, but cells are the basic fundamental unit of living beings. They either exist as a single cell or a combination of multiple cells. They either exist as single cell, say unicellular, and the combination of multiple cell, that is called multicellular. Now we discuss discoveries about cells and the fundamental unit of life. First discovery is a patent by Robert Hooke in 1665. He used his crude microscope to find the first cell, and these are the dead cells of cork of slice. These are the dead cells of cork of plant, that is the outer membrane of the plant bark, that is cork, and they are the look like a honeycomb projection. And cell word is derived from a word, Latin language cellula, which means a little room. Next discovery is patented by Antony von Leeuwenhoek in 1675, about after 10 years of Robert Hooke. 1675 he make a advanced microscope which is used to find the first living cell. He found the presence of first living cell using pond water, and he also discovered the cell of sperm of dogs, sperm of frogs, sperm of humans also, and RBC of human. After that, Robert Brown in 1831 recognized the existence of nucleus of cell. He said that cell has a center part called nucleus. Existence of nucleus of cell is discovered by Robert Brown. Then J.E. Purkinje in 1839 invented the term protoplasm, which is the liquid present inside cell. Purkinje tells that the total fluid which present inside cell, that means including nucleoplasm and cytoplasm, both are combinedly called protoplasm. Then Matthias Schildian and Theodor Schwann in 1838 and 1839. Matthias Schildian is a botanist and Theodor Schwann is a zoologist. They both proposed a cell theory. That means all organisms are actually made up of cell. That tells us every organism is made up of cells, and some more fundamental points of it. Then after that, 1855, after Matthias and Schildian, Rudolf Virchow came in existence, and he modified the cell theory. And suggests that all cells come from a cell that already exists in nature. That means a simple say cells are exist from pre-existing cells. And that means any one or living organism are originate from their pre-existing ancestor or cells. Now some common point and useful point related to cell theory. A cell is a structural and functional unit of living organism. All the living organisms are made up of cells, and cells are originate from pre-existing cells. Now we discuss about unicellular organism and multicellular organism. The organism that consists of a single cell, such as amoeba, are called unicellular. In unicellular organisms, all life functions or processes like respiration, nutrition, reproduction, etc. are performed by a single cell. In multicellular organism, which contain various cells that perform the different functions in the organism, such as a plant, fungi, and animals. So multicellular have the more than one cells, they have the different function. That means different division of labor. They have a division of labor. Like RBC have a function, red blood cells have a function to carry oxygen and supply the oxygen to the each and every part of a body and carry out the carbon dioxide out of from the blood. So and white blood cells have a working to protect against the upcoming pathogen or produce antibodies. So this is a different function of different cells. This is called division of labor. This is called division of labor. So first question arises here is, how can multicellular organism originate from a single cell? So a cell has a capability to divide itself into cells of its own type. This is known as cell division or cell reproduction. Therefore, the more cells can generate from already existing cell. तो हम कह सकते हैं more cells are exist from already existing cell, and this is known as cell differentiation. When the one kind of the cell form the another kind of the cell, this is known as cellular differentiation or cell differentiation. For example, in human, number of the types of cells are originate from zygote. We all know that during the fusion process of sperm and ovum, zygote is formed. Sperm and ovum mix together to form zygote, and zygote is a single cell, and we are originated from a zygote, and number of kind of cells are formed from a single zygote like RBC, WBC, platelets, bone cells, etc. are originate from a single zygote cells. So this is called as cellular differentiation or cell differentiation. Next point is shape of cell. The shape of cell may vary depending upon the type of function they perform in an organism. The shape of the cell may vary depending upon the type of function they perform in an organism. जो shape है cell की वो vary करती है, depend करती है function they perform by organism. क्या function उसके अंदर करेगी. Cells are capable for changing their shape. For example, cells are capable for changing their shape like white blood cells and amoeba can change their shape their own. अपनी shape को खुद से change कर सकते हैं because they have a function of their shape change. Like amoeba capture their food by change their shape and locomote with the help of change their shape. And white blood cells cover the pathogen and kill the pathogen and pass from the narrow passage of the blood vessels due to change their shape. Xylem vessels, like in plants, example of plants, xylem vessel in plants are the long as they have to form long water conducting channels. They form the long water conducting channels, and we know that xylem tissue is that which present in plants help in transportation of water. Next is, how can cell perform distinct function in an organism? Cells are capable of performing multiple functions in an organism. A cell contains specific components which called organelles. A cell contains specific components called organelles. Each organelle in the cell can perform different functions such as making of new cells or clearing the waste of the cell. Clearing the waste of cell. Thus, organelles allow a cell to perform several kinds of activities in an organism. Thus, organelles allow a cell to perform several kinds of activities in an organism. Okay, and after that chapter, after this topic, we discuss the each and every cell organelle and their function. Now, organization of cell. A cell contains mainly three organization sections: plasma membrane, nucleus, and cytoplasm. Plasma membrane, nucleus, and cytoplasm. What is plasma membrane? It is just like an envelope that covers the whole cell. It is just like envelope that covers the whole cell. Therefore, a cell gets separated from the external environment because of its plasma membrane. You know that cell has the outer boundary. This boundary is known as plasma membrane or cell membrane. The plasma membrane has a capability to decide which material should enter or leave the cell. The plasma membrane have a capability to decide which material should enter or leave the cell and which should not. That's why it is called selectively permeable membrane or SPM. That means it can pass through them selectively particles. That's why it is called semi-permeable membrane. Now we discuss the fluid mosaic model of plasma membrane. It is a microscopic model which tell about how the plasma membrane structure is made up of. Fluid mosaic model explain the structure of plasma membrane. Fluid mosaic model explain the structure of plasma membrane. According to the plasma membrane comprises the three components. Plasma membrane is made up of lipid, proteins and carbohydrates. Lipids, proteins and carbohydrates. These components can follow freely and fluidly inside the plasma membrane. ये जो component है freely और fluidly फैले रहते हैं, उसके अंदर तैरते रहते हैं. There are the two types of lipids or fats in the plasma membrane. Phospholipid, it is a lipid made up of glycerol. Glycerol is an organic compound and two fatty acids attached with them and one phosphate molecule. It creates a semi-permeable membrane which allows to flow any certain material inside or outside. So phospholipids are responsible for to carry the certain material go in and out of the cell. That means they allow the material in and out of the cell. Now what is the working of cholesterol? It is a lipid which provides fluidity to the surface of plasma membrane. It provides the fluidity to the surface of plasma membrane, and the protein act as a receptor. Third is proteins. Protein act as a receptor of the cell and help in transportation across the cell. They help as a receptor membrane, and carbohydrates attach themselves with the lipid and proteins and are the found on extracellular side of membrane. This is plasma membrane model, fluid mosaic model of plasma membrane. These are the known as phospholipid molecule or lipid chains, which have the one end is hydrophilic and second is hydrophobic. Hydrophilic is that this is known as phospholipid bilayer. Hydrophilic, that means water loving. Hydrophobic means water repelling part. Water repelling part. So they allow the material to go in and out of the cell. And during active transport, the materials go from lower concentration to higher concentration. But in generally, they are moved from higher concentration to lower concentration. How can a substance move in and out of cell? First is gaseous exchange between the cell and its external environment. The movement of gases like oxygen, carbon dioxide to and from the cell is carried out by a means of diffusion. So diffusion is occurred, and we know that how the diffusion according to concentration gradient, diffusion is from higher concentration to lower concentration. Movement of diffusion, higher concentration to lower concentration. Gaseous substances have a tendency to move to area where the concentration is higher to area where the concentration is low. Now movement of the water between the cell and its external environment. Then the liquid is moved from higher concentration to lower concentration around a cell membrane, across a cell membrane, then it is called osmosis. Osmosis is a special kind of diffusion process. Now we discuss the three kind of solution on basis of osmosis. First is hypotonic solution. The word hypo means less concentration. That is, concentration inside the cell is less. That is means hypotonic medium is that medium in which concentration inside the cell is less and it can absorb liquid. Concentration of the liquid inside the cell is less, then it can absorb liquid, and generally the liquid is water. So cell gets swell up, and movement of liquid from outer medium to inside the cell gets swell up. Next is isotonic solution. If the cells are put in an environment which has the similar concentration, similar water concentration present inside cell, that means both have the equal concentration. The word iso means equal, and concentration in and out of the medium has the same. That is, both are in equilibrium. No actual exchange takes place. No movement takes place, no swelling happen, no shrinking, no swelling. Next is hypertonic solution. Hyper means extra. Hyper means extra or more. If the concentration inside the cell is more than the surrounding medium, then shell starts to shrink and the liquid which present inside the cell comes out from the cell. This is known as hypertonic solution. I hope student you understand the above section which we have discussed. Isotonic, hypertonic and hypotonic solution. Next is endocytosis. A process by which a plasma membrane engulfs food and other material inside the cell is known as endocytosis. Now endocytosis is three types. First is phagocytosis, when a plasma membrane engulfs a solid particle, that is known as phagocytosis. If it is extract the liquid particles or fluid particles, then it is called as pinocytosis. If it can accept the receptor mediated endocytosis, when they accept the any foreign particles or pathogens, then it is called receptor mediated endocytosis. Next is what is cell wall. A cell wall is the outer hard covering of the cell which maintains the shape of cell. A cell wall is outer hard covering of shell which maintain the shape of cell. The cell wall is generally made up of cellulose. In the plant, cell wall is made up of cellulose. In bacteria, made up of peptidoglycan, and the fungi or mushroom made up of chitin. Now, next question is, what is plasmolysis? Is it a process in which the contents of the cell that are away from the cell wall shrink or contract when the cell loses the water due to osmosis when it kept in the hypotonic medium? Hypertonic solution, when it kept in hypertonic solution. अगर इसे hypertonic हम solution में रखते हैं, then it can remove the whole water. It can remove the whole water at cytoplasm getting shrink, and cell wall does not change the shape. This process is known as plasmolysis, and cell is plasmolysed. Another one is isotonic. When we keep this cell in isotonic medium, then equal concentration and cell shape do not change. That is called flaccid cell. That is called shape is flaccid. Third one is turgid. When it can absorb water but does not blast, does not burst and completely fill the liquid and all spaces are completely filled, then it is called turgid. Then it is called turgid. And shape is called turgid when we kept in a hypertonic solution, then it is happened. Now question is, can the dead cell absorb water? No. Dead cell cannot absorb water through osmosis because have no cytoplasm and no concentration gradient there and no exchange takes place. How the plants, fungi and bacteria exist in hypotonic medium? Plants, fungi and bacteria exist in such situations because their rigid cell wall help to make it turgid, to become turgid, but it does not burst out. वो फटेंगी नहीं but becomes turgid हो जाएंगी, hard हो जाएंगी, as you see in this figure. Now we discuss the structure of plant cell and animal cell. First we discuss the parts of any plant cell. Plant cell is a outer hard covering known as cell wall. Then the inner membrane which attach with the cell wall is cell membrane. Golgi apparatus, or they are called as not improved Golgi apparatus, they are called dicytosomes in plant cells. These are chloroplasts which contain chlorophyll, and Golgi vesicles, the vesicle-like structure, the ball-like structure. Ribosome, the granular-like structure which present in it. Smooth endoplasmic reticulum which contain no ribosome over it, and rough endoplasmic reticulum which contain ribosome over it. And the larger vacuole present in plant cell. This is vacuole membrane and vacuoles are present. And this is nucleus, and nucleus have the center part is called nucleolus. Rough endoplasmic reticulum, large central vacuole, and amyloplast starch gene. That means the starch molecule called amyloplast. And the most famous is mitochondria, and the fluid present inside is cytoplasm. And we discuss each and every term one by one in next section. Now we discuss the parts of animal cell. Pinocytic vesicle, that means they are looking like a pin and help to enter the liquid or other material inside the cell. Mitochondria, Golgi bodies, Golgi vesicles, lysosomes, nucleolus, nucleus, SER, RER, centrioles. So centrioles are that organelle which does not present in plant cell, and the plastids or chloroplasts are those organelle which are not present in animal cell. Microtubules, cytoplasm, ribosomes, or cell membrane or plasma membrane. I hope student you understand this one section of the chapter. Thank you and have a nice day. Stay home, stay safe.